So our next speaker, and the final one in this session today, is a professor at University of Ottawa in the Faculty of Law, and he's co-founding director of the Open African Innovation Research Network, Open Air Africa. He's also a senior fellow at the Centre for International Governance and Innovation and senior research associate at the University of Cape Town. He's got a very distinguished background, and I'm very pleased to welcome him here today to give a presentation on gene editing for innovation in agriculture, global implications. And it's Jeremy De Beer. Thank you.
I'm going to speak specifically about CRISPR as a particular gene editing technique, and talk a little bit about the implications for global agriculture innovation and food security. Now, my area of geographic focus is on Africa. As Peter mentioned, I'm a co-founding director of the Open Air Partnership. That's the Open African Innovation Research Partnership, funded by IDRC and SSHRC and numerous other donors. It's a partnership between universities in Canada and 15 countries on the African continent. We've been working for over a decade on innovation, looking at regulatory systems, intellectual property, and in a way, overall open science systems. And that's central to my point today, that I believe gene editing is a revolutionary technology, not only for its scientific merit alone, but for the way in which it's going to reshape open science systems. So I'll start with an answer to the question that was asked about gaining acceptance for new technologies on the African continent. And I think that CRISPR has a tremendous potential to do that. And the reason is because this is a technology that is likely to democratize gene editing. It's going to make the process more inclusive and more participatory for a few simple reasons. It's cheap and it's simple. Being cheap and simple, CRISPR systems will change the way that plants are genetically engineered in the short term. In the long term, it'll change the way in which all organisms are genetically engineered. Have you spoken already about CRISPR specifically? Would you benefit from a little bit of a primer? So the acronym CRISPR, which is clustered regularly interspaced short palindromic repeats, were first identified in the late 1980s as present in a bacterium or bacteria as essentially a way of coding for viruses that bacteria have encountered. Think of it as a genetic vaccination card. Every time that a bacteria encounters a virus, it takes a snippet of the DNA and then is able to identify that so that not only that organism, but progeny of that organism can identify the virus, remove that from the organism, and knock it out. So when we think about the way that gene editing used to work, analogize it to the printing press, an early printing press, where in order to do gene editing, you had to assemble all of the letters that you wanted to print. You had to do it manually by hand, and God forbid there was a typo in that process or anything went wrong, you'd have to recreate the thing all over again. CRISPR is essentially the find and replace function in Microsoft Word compared to the printing press. That's the difference. And in the same way that that computer software has revolutionized everything from every aspect of information communications technologies, CRISPR's already doing the same thing in biotechnology. Okay, so because of its simplicity, actually, let me just back up. There's a few other aspects. So not only does it have the simplicity of the find and replace function in a word processor compared to manually setting a printing press, it's significantly cheaper. So in order to do genetic engineering using conventional technologies, you would have to purchase materials in the range of $5,000 would not be an unreasonable price to pay for the materials you need to genetically engineer a crop. just once, to do one experiment. Comparably, you can get everything you need, the rDNA and the Cas9 protein, to use CRISPR systems for about $30. So that's the price comparison we're talking about, $30,000 to $5,000. In addition, the final thing is it's far more accurate. When you're using conventional genetic engineering techniques, I realize that sounds like a little bit of an oxymoron, but conventional genetic engineering techniques, there's a high margin of error because essentially to oversimplify, when you're inserting the new genetic material, where exactly it gets inserted is by and large a random process. But using CRISPR systems, you can be incredibly precise. You can deactivate, knock out, or replace very specific sequences. So I think overall, the potential here is to move genetic engineering outside of universities and large-scale private companies and publicly funded research organizations into the hands of citizen scientists. And when we see citizen scientists adopting these technologies in a do-it-yourself way, in biohacker spaces, we're going to see a grassroots acceptance of the technology in a way that we don't see with more formalized top-down systems. Now, there are two kinds of networks that I'd like to speak to you about that are going to be relevant to the adoption of CRISPR systems in genetic engineering, both for human therapeutic use and for agriculture and food and biofuels and industrial bioprocessing. The first thing I'd like to talk about is some of the licensing networks, the intellectual property landscape and licensing networks that are evolving around CRISPR systems themselves. And the second is related to the way in which CRISPR and gene editing fits into the science ecosystem or the open science ecosystem, including open access, open data, do-it-yourself biology, synthetic biology, and more. So let me talk a little bit about the licensing networks around CRISPR in the first place. CRISPR wasn't really a thing that was in mainstream consciousness until about 2012. And in 2012, two scientific papers were published. The first paper to be published was released by a professor at the University of California, Berkeley, Jennifer Doudna, and a co-author from the University of Vienna, Emmanuelle Charpentier. Now, these two women were the first women to publish on CRISPR, and they published a paper that explained how you can use this system that I've described in prokaryotes, in essentially bacteria cells that don't have a nucleus, for simplicity's sake. So this was the application of the general idea of how you could use this technology in the most basic setting. And the paper described this as a tool for gene editing. So they were working on particular applications and uses of this tool, but they described the tool generally. Now, a few months later, a paper was published by a researcher at the Broad Institute. That's a joint venture supported by MIT and Harvard. And his name is Zhang. He's a young scientist. He was 34 when this was published, and is now sort of quite well entrenched as the leading inventor on patents and has the most publications on this. And what was unique about this particular paper was that he described a way to use CRISPR systems in more complex organisms, in cells that have a nucleus, like plant and animal cells and human cells. So these papers were published, but not before the university tech transfer offices did their job properly and filed patent applications. So there are two competing patent applications put in. One jointly by the University of California, Berkeley, and the University of Vienna. And the other by the Broad Institute, MIT, and Harvard, so sort of the West Coast group and the East Coast group. Now, the priority date, which is the all-important magic point in time that you get to claim priority for your patent application, for the West Coast group, well, I guess Vienna's not on the West Coast, but anyways, the Berkeley group, the Berkeley and Vienna group, Doudna and Charpentier, was May of 2012. Now, the other group, the East Coast group, the Boston conglomerate, Broad Institute, MIT, and Harvard, their priority date was December 2012. So under most systems in the world, at all systems in the world now, it's not the first person to invent a technology that gets the patent lottery, it's the first person to file the application disclosing their invention. Except in the United States, at that time in 2012, it was a different patent system. It was the first to invent system. So it opens up a big evidentiary can of worms about who came up with what when. Now, although the priority date for the Broad Institute and MIT and Harvard was December 2012 compared to the Berkeley-Vienna group's priority date of May of 2012, the Tech Transfer Office at the Broad Institute did something very clever. They paid an extra $70 for what is known as an expedited review of their patent application. So having paid the $70 for an expedited review of their patent application, that patent was in fact the first to be granted by the United States Patent and Trademark Office. Now, that is a big deal, a really big deal, because once that patent has been granted, even though there was the literature on the books and the other application in the stack of papers to be processed, the University of California at Berkeley and University of Vienna alleged that that grant of that second patent to the Broad Institute, quote, interfered with the prosecution, the consideration of the Berkeley patent, Berkeley-Vienna patent. And so they brought proceedings under an administrative process known as what's called an interference proceeding, and they essentially alleged that the MIT Harvard Broad Institute group ought not have been granted the patent because the information was already known as a result of the earlier paper. And that if the Berkeley-Vienna group had disclosed a method of using CRISPR systems in simple organisms without a nucleus, that it was obvious that the same system should be applied, should be tried, and would work in more complex organisms. At the first decision, the United States Patent and Trademark Office ruled against the University of California, Berkeley, and Vienna, and in favor of the Broad Institute. So that patent currently stands. There was an appeal hearing where arguments were made last fall, and a hearing was made just in April, I believe, of 2018. Although on appeal, it's going to be really hard to overturn that finding. So what's been happening meanwhile? A couple of things in the patent landscape. Meanwhile, UC Berkeley and the University of Vienna have been working to build up a more robust portfolio, trying to file a few other patents, and they've got a few dozen. But the Broad Institute has gone berserk. They have been patenting all kinds of stuff around this environment and around this space in association with their partners. And that's a really good strategy because, quite frankly, nobody knows what's going to happen with these original core patents. And so it's creating a lot of confusion in the marketplace about who to license from. So even if you wanted to get into the CRISPR business and you said, oh, this is great, I got to get in, and I want to do things on the up and up and get a license, you're going to go into your intellectual property lawyer's office and she's going to say, well, the problem is you don't quite know who to take a license from. Right now it's a horse race. You've got to bet on a particular horse or get to a strategy I'll talk about in a second, which is a very clever one. And so there's a lot of confusion as to who you should take the license from. Now, I don't know if you remember it's about the same time, 2012, there was some patents on two genes, BRCA1 and BRCA2, by a company called Myriad Genetics. And these patents, essentially mutations in the genes identified by these patents, were linked to an increased risk of breast or ovarian cancer. And a company called Myriad Genetics in the US said, no, we own those genes. And the United States Supreme Court said, no, you don't. You can't own genes. But by the time that the Supreme Court issued its ruling, Myriad had actually built up such an other array of patents around the core technology that when you knocked out the sort of legs from underneath it, everything else managed to kind of hold together a little bit, and they were able to maintain their competitive position. That's what the Broad Institute's doing now. Also, meanwhile, what happened was another player came out of the woodwork. It turns out that a university in Lithuania, Vilnius University, which is a large, prestigious, and very old university in Eastern Europe, had researchers working on this same stuff all along. Except they weren't issuing press releases in the US media about it. But meanwhile, lo and behold, what did they do? But they acquired a portfolio of European patents over the core technology, and in fact now have one of the strongest positions. Okay, so those are some of the key players behind the technology themselves. Now, as you would expect, the entrepreneurial universities, being what they are, have spun out a number of startups which have literally hundreds of millions of dollars in seed funding, are mostly around medical therapeutic uses. That's where people think the real money's going to be in the long term. That's where the venture capitalists are gravitating. But there's a really smart player in all of this, and I'm sorry that I missed the presentation earlier, but that's Dow DuPont. And Dow DuPont has been really clever here in this space because they recognized that one of the first uses of CRISPR systems was actually in the yogurt business. What was happening is you could use CRISPR systems to essentially eat up lactose in yogurt, and there was a Danish yogurt company that was acquired by DuPont, and prior to its merger with Dow, now has slowly but surely acquired the largest and most, I would say, secure stake around the CRISPR patent landscape of any company other than the originating universities. It's the main private sector player. It's not the only private sector player, but definitely the main private sector player with the strongest hand in this space. Now, why is that clever to go into the agriculture and food sector? The reason is because it does not trigger the massive ethical questions that exist when it comes to human therapeutics and the use of CRISPR in human or animal populations. Now, here's why. One of the very important things about CRISPR is its potential to do what's called a gene drive. Have you heard of a gene drive before? Gene drive, where it's a technique where you can very quickly get a genetic trait through an entire population in 2 or 3 generations, or even less, because the gene is you can mate 2 organisms where the gene is present in one and spliced into the other, and all of the progeny within 2 generations have that characteristic. So you may have heard about this idea of eradicating mosquitoes in order to fight malaria. The idea was to use CRISPR technology, CRISPR systems, to do that. In China, scientists have used this on human embryos, although they're quick to point out never implanted. But the potential exists for major or potential eugenic implications. When we talk about designer babies, all of this is very easily facilitated by CRISPR. So some scientists are actually calling for kind of a moratorium on R&D in this space because of the massive ethical questions. As a little parenthetical or an aside, that's one of the big questions that's going to emerge around citizen scientists, right? When university researchers have research ethics board and corporations have shareholders and the media holds these organizations to account, or the civil society holds these organizations to account, there's a real check on what can happen and what can be done ethically. But once this gets into the hands of citizen scientists, those checks and balances in the research ethics system really go away. So that's just as an aside. So, however, in respect of food and agriculture systems, we don't see the same concerns emerging. And in fact, we see the opposite. So the U.S. Department of Agriculture just announced formally the policy that it will not regulate CRISPR-engineered plants the same way as other transgenic or genetically modified organisms. It will not. So green light in the U.S. Australia is highly likely to follow suit. In Europe, where we might expect sort of the most hostility, the tide is turning. In Europe, there's a directive, the 2001 GMO Directive. There was a group, a civil society group in France, Confédération Paysanne, that brought a challenge under French law, arguing that the EU directive, the GMO directive, applies to CRISPR-engineered plants. The Court of Justice hasn't ruled, finally. However, before the Court of Justice issues a ruling, there's a process known as an Advocate General's opinion. It's essentially like an advisory opinion. And 9 times out of 10, the Court of Justice follows the Advocate General's advisory opinion. And that was released in January of 2018. And the Advocate General advised the European Court of Justice that the GMO directive in the European Union does not apply to plants modified using CRISPR systems. And the reason, in short, is because they're not transgenic organisms. You're not taking genetic material from one organism and putting it into another organism. You're only modifying the organism's own genome by activating or deactivating certain genes. So that falls under what under the EU directive is called the mutagenesis exception. We expect a ruling literally any day from the European Court of Justice. But if the US and Australian approach and the Advocate General's approach in the EU holds, we've got green light for CRISPR-engineered plants for food and agriculture that will face none of the barriers that conventional genetically modified organisms face. No risk assessments, no traceability requirements, none of those problems. Okay, so that brings me to the sort of broader systemic implications of this technology. And there's a little bit of a paradox here, because the intellectual property landscape is becoming so crowded with the stories that I told you involving UC Berkeley and University of Vienna and the Broad Institute, MIT, Harvard, Vilnius University, the various spin-offs, Dow DuPont. And it's fascinating to see all of the venture capital focus on that sector. I don't believe that in the long term that's where the action's going to be. At best, the patent lasts for 20 years. So those original inventions will be expired in 2032. And that sounds like a long time away, but it's not. That's right around the corner. 2032 is right around the corner. And I think by that time, what we will see is the widespread adoption of this technology by citizen scientists. And what happens in that environment is citizen scientists don't manage intellectual property risks the same way that small and medium-sized enterprises, let alone large corporate enterprises or universities and publicly funded research organizations do. It's a much more, I want to say cavalier, but maybe entrepreneurial is a less pejorative term, a much more sort of just get it done culture that we're seeing in citizen science. And once the cat is out of the bag here, I think we're essentially looking at, this is like the Napster moment of genetic engineering is about to happen. And once that's the case, we're not going to be in a situation, I would predict, where the core patents are going to be broadly licensed by a lot of people, a significant number of people who are using these systems and these techniques. So I think the real action, and this is what the venture capitalists are really interested in as well, the real action is on the periphery. It's specific applications of these techniques to generate breakthroughs in human therapeutics or food and agriculture or biofuels or other industrial applications of biotechnology. So I think what we really have to do to understand CRISPR and its big picture implications is position this alongside some other megatrends that we're seeing in science systems. I think it's really important that we understand this new technology alongside do-it-yourself biology and biohacker spaces and those citizen scientists that I've been talking about. And that group, that constituency of actors in the science system are fueled not only by these new technologies, by CRISPR, but also the rise of open agricultural data and the kind of information that the PERC project is producing. Once we have these massive databases of agriculture-related information, whether it's field test data or meteorological data or commodity pricing data, all of those open data systems are also feeding into this system. We're seeing fundamental changes in the publishing and scientific publishing industry, where we no longer see the barriers to accessing scientific literature that we once did. I don't know if any of you are familiar with websites like Sci-Hub. This is the Russian pirate site where you can literally get any scientific article you want with simply the DOI, and trust me, it's easier to use than Scopus. So all of this, I think, fits into a much broader change that we're seeing in science systems, and CRISPR is really just a part of that. So that has radical global implications, I think, not just for us sitting here in a developed world institution, sophisticated center of plant breeding excellence, but it's going to really democratize and decentralize power structures. I think what we're going to see in the medium term, not in the next couple of years, but in the next, say, 10 to 15 years, is a possibility for entrepreneurs, particularly in developing countries that haven't had the capacity to do big science, take a role here, and there's a real potential to leapfrog what's going on in OECD countries because of the more flexible regulatory systems. So in the same way that we've seen things like mobile money take off in the absence of banking regulations in the African environment, we may be able to see DIY biology take a leap ahead or around the regulations and the ethical constraints that are more entrenched, I think, in a place like the University of Ottawa or the University of Saskatchewan or Berkeley or the Broad Institute. If you're interested in reading some more about the Broad systems, I've offered a couple of links to additional reading that I've produced with team members I've been working with on the PERC project and here at the Global Institute for Food Security in partnership with our Open African Innovation Research partnership. And all of that work wouldn't be possible without the generous support of our funders. And let me just end by acknowledging that support. GIFS and PERC in particular, supported by IDRC and SSHRC and the Canada First Research Excellence Fund.